All right, guys, so for this video, I wanted to get into genetics a little bit. Obviously, we can't cover everything in one video because there's a lot of things there to cover. Also, just for this, we're going to isolate it to one species. We're just going to talk a little bit of blood pythons. I have 4 different blood pythons to show you. 3 are siblings, 1 is from a separate pairing. But they all involve some genetics that I'll be able to show you some differences. So also, last time with certain words and things like that, my pronunciation was not so great on one, and other things you might just miss the concept. So I made these handy cards for you so I can discuss, a trait, for instance. So it seems like a simple thing, a trait, your hair color, your eye color, all these different characteristics that make you can be traits, and those can be inherited genetically. So you could have brown hair, you could have blonde hair, and these things are predetermined on several levels. So in understanding how this stuff works, probably one of the most important things we want to learn about are going to be chromosomes. Now we know in people we have 23 pairs of chromosomes, or 46 chromosomes. We inherit 23 from our mother, 23 from our father. Well, snakes don't have the same number of chromosomes that we do. And snake species have different ones. And I'm going off the top of my head, so I may be wrong. I forget which is which, but like berms and corn snakes, one of them has like 74, one of them has 78 individual. So 37, 39 pairs. So there's a lot of different chromosomes involved in them as well. It's just a little bit different on the numbers. And so people might ask, what is a chromosome? It is an entire chain of DNA that gets, along with a group of stabilizing proteins, and it's in this chromosome here. And what really affects the genetics that we tend to look at, another handy card here, something called alleles. And alleles, it's a different form of a given gene in a specific spot on a chromosome. And so I made a visual kind of for that too. We're gonna get a little ahead of ourselves. We're gonna talk about, these things called linked traits, which is not necessarily all that common in snakes. I've heard some rumors of this one's linked or that one's linked. So we're gonna pretend that these paper towel tubes here are chromosomes. This is not what a chromosome shape looks like. Chromosomes kind of look like a unwound ball of yarn, except when cells are dividing and things are going on, they actually look like, your wrapped brand new thing of yarn. So you have these two, and you can see the hair color on here is what I used. Those are your alleles. So those alleles are attached to the chromosome in a specific place, and they may not be perfect because I'm not perfect, but in the real world they would be. So when a parent has this pair, one or the other of these is going to go to the offspring, and therefore the genetic information on this chromosome and within this allele is going to pass to that. So in this case, if, I don't know the dominance and recessive with hair color, I believe brown would be dominant. But say this person's, brown hair, but they carry the genes to make blonde or however it works. So you would have, the information here to make that. So depending on what passed, which is where you get into your Punnett squares, and hopefully everybody knows what Punnett squares is when you're dealing with Mendelian genetics. If this one lines up, this could be that recessive blonde, whereas this one could be that dominant brown. And that recessive blonde's never gonna show unless the other pair has that recessive as well. But now with linked traits, what happens here is I have this other set, and this set has eye color on here on these chromosomes. But if you look down here, we have nose size. So now with linked traits, when this chromosome or that chromosome goes into the pairing, automatically those two things go together. So you won't ever separate them. You may not visually see those things, but the genetics to make them will always be there. I know at one point in ball pythons, it was thought that hidden gene loma and granite were linked. I don't know if that's been proven false or true, but it did seem to occur a lot more often where you had, once you put those traits together, they stayed together. I don't know, like I said, I didn't really breed those genetics in balls, so I'm not sure. But if that it did work out true, that would be an example of linked. Once they're linked, You're not going to separate them. So we talked a lot about, what you're going to see versus what you're going to not see and all that. So that's where these terms you hear of homozygous and heterozygous come into play. So homozygous, if you break it down, homo means the same. Zygous is the mix of DNA that you get. So homozygous means you're getting the same, or you're showing the same DNA. Whereas heterozygous, hetero means different. And so in that case, you've got something going on where you're looking and you're not necessarily seeing the homozygous form of the gene. You're seeing a heterozygous, which means that it's carrying the information to make that, but that it's not necessarily manifesting visually. And in some cases, it may be depending on the type of gene we're talking about. If you watch the video, we talked about the difference between codominance and incomplete dominance and all that. That comes into play. So now along with those terms is genotype and phenotype, which is another thing you probably hear a lot. So what is the difference? The genotype is the genetic constitution. It's what's actually in there. You don't always necessarily see the genotype, but that means it's in there. So when we're dealing with an animal that's heterozygous, its genotype is going to have the gene to make, let's say it's albino, which is a simple recessive trait. It's going to have the gene to make albino, but the animal's not going to be albino. So its genotype contains that. But the phenotype, in that case, this is what you actually see. So you're going to see a normal animal, but the genotype is going to carry the genes to make that albinism. And so now we'll get into showing you some animals and kind of how this applies. So to start, I'll do the three siblings here. And the pairing in this case was a batik blood python that was heterozygous for T-positive albino, paired with a visual T-positive albino female. So the two genes are different. Albinism, as I said, is a simple recessive. So if you breed two albinos together, you're going to get all albinos. If you breed an albino to a non-gene carrier, just a normal or wild type animal, you're going to get all normal looking animals. So the phenotype is going to be normal, but the genotype, they're all going to carry the gene to make albinos. So they're all going to be heterozygous. So you have the homozygous albino going to the normal or wild type, resulting in heterozygous offspring. Now, the batik works differently. That's an incomplete dominant gene. So you breed a batik to your normal or wild type, you will get batiks in that first breeding. They won't all be batiks. It works kind of almost like a het, where half the babies get that or breeding a visual to the incomplete dominant kind of and recessive are almost the same trait, other than the fact that the incomplete dominant, that gene, when it's the het form, has a visual effect on the offspring. And so that visual effect is affecting the phenotype, whereas the simple recessive doesn't affect that. So that's the difference, and that's why even though those genes, from a technical standpoint, as far as odds go and all that, work exactly the same, that's why they're categorized differently. One affects the heterozygous offspring and the other doesn't. Now that doesn't mean that a recessive trait cannot affect the phenotype of the heterozygous offspring. Granite Burmese pythons, for instance, a lot of het granite burms will have that speckling similar pattern to a granite, a little bit reduced, busier, whatever it is, but it's not a guarantee that happens every single time, and so therefore it can't be labeled incomplete dominant, it has to be labeled recessive. So the genes work essentially the same way. The biggest difference is how the phenotype of the animal is affected. So as I said, we have these siblings here from the same clutch. So the pairing again was a batik het for T-positive albino. So he's visually showing the batik gene, and he visually looks normal colored. You don't see that albinism, but he carries the genotype to make it. And then the mother was a T-positive albino, so her, phenotype, she's showing that recessive trait. It's in its form. So this baby here from that clutch appears normal, you know, your wild type. But this baby carries the gene to make albino. So we call this het albino or heterozygous for albino. So this baby, when it's bred to an albino, you can get half albinos, half are going to be gene carriers. If you bred it to another het albino, you'll get a quarter visual albinos. A quarter babies won't get that gene for albinism anywhere, and then half of the offspring will be gene carriers, just like this one. Then if you bred this one just to a normal wild type, you won't get any visual albinos, but half the babies will be like her here and be gene carriers. Now, also with these snakes, another thing that's going on here that we didn't discuss is line breeding. And so in line breeding, it's not a cut and dry scenario where, you know, these genes go in and you're going to get a predictable result. Line breeding, you're breeding for color in this case. I want redder snakes, and these snakes take several years to get to their adult colors, but this girl hatched out very brown, and it doesn't really show up, but she's already very red, and she's going to be really, really nice. I don't know where she's going. I don't think she does either. So with the line breeding, you're picking animals that's phenotype is appealing to you, and even though there's no necessarily, no genetic you can really pinpoint, if you match them up over generations, you can produce more predictable results. So that's line breeding. And line breeding is kind of a fancy term for inbreeding, essentially, because typically you start off with siblings that have that desired look, and you try to keep heading down in that direction and keep that line tight for a while to reproduce the results you want. And then at some point, you start outcrossing, which you should look to do as quickly as you can while keeping the integrity of what you're after. So again, she carries the gene to make albino, but visually she's a normal or wild type. So now we'll grab one of her siblings who hopefully is going to be cooperative here. Hold on, let me switch up some things because, working with animals. Bear with me a second. He crawled right through the camera tower here when I went to grab him. He just got stuck. All right, so this female that I'm grabbing is this snake's sister. So they are siblings from the same clutch and the same parents, but this one is T-positive albino. So she's the visual albino. This one is the heterozygous albino. So what happens is with those alleles we talked about is one copy has the albino gene in this one, whereas two copies have it in this one, which is why she's visual. Both alleles have that albinism, whereas this one, just one, has that trait. And so that doesn't show. And it's the same in the incomplete dominant traits that we talked about, where, let me put them down for a second, and I'll grab the third animal. I know, it's a tough life. No, you have to stay in the bin. That's how this works, because I can't. Oh my God. They just do not want to cooperate. Okay, so now this guy here is also a visual albino. So he has both copies, both alleles have the albinism, but he's also batik. And so, as I said, batik is an incomplete dominant. I don't have a super batik, but they're basically a patternless blood python that are kind of I don't know what color you would call it. It's kind of a creamy tan color, but all that pattern would be gone. So this is sort of the het form, if you will, of that incomplete dominant gene. So while he's fully expressing both alleles of the albinism, he just has the one allele of batik. And so there's different things going on. And once again, you have that line breeding in here, which is what gives him his color and why he's very red. Like I said, they get washed out indoors. So there's a lot of things that you can have going on in one snake. And of course, it makes it more and more complicated as you layer stuff on, depending on the types of genes and the outcomes and the lineage that you're working with. So that's why it's important, especially with blood pythons and short tails. A lot of people care about the lineage, because we want to know what we can expect when we produce offspring with these animals. Not only just the cut and dry stuff, like this animal's going to produce more batiks, this animal's, able to produce, offspring that are either heterozygous or homozygous albino, but also the lineage of the red. And we can look back at his parents and grandparents and see the red that's in there. And then from there, we have an expectation of what direction we can head and what we can expect from his offspring. So the last baby I'm going to show you is also going to be albino, T-positive albino again. But he's a T-positive albino ivory blood python. And so in this case, the ivory gene is similar to the super batik, or the batik and the matrix are the same. So matrix is your heterozygous form of ivory. That's an incomplete dominant. So I didn't grab a matrix for this video. Let me see, he looks a little out of sorts. Haven't had him out much lately. So this is a T-positive ivory blood. And so what's going on with this snake is not only does he have both copies of the albinism, to both alleles, he also has both alleles for ivory. So he has the super form of the incomplete dominant gene, as well as, the homozygous form, if you will, and the homozygous form of albinism. So with this snake, everything that you breed him to, if I just bred him to a total wild type with nothing going on, every single baby will be matrix and every single baby will be a gene carrier for albino, because they're both going to get one copy of those alleles. So you're going to get matrix, which is the visual het for ivory, and then you're going to get that heterozygous for albino. Now, if I breed him to an albino, I would get all albino matrix. If you breed two visual albinos together in T-positive blood pythons, every resulting offspring is going to be albino, which is how a simple recessive works. And because this is the super form or the homozygous form of that ivory gene, you can't create normal offspring with him because the matrix gene always alters the phenotype of the resulting offspring. So with matrix, it can alter the pattern. That's not guaranteed every time, but what is that the tongue color changes from black to pink. So in blood pythons, if you have one with a pink tongue and it's not albino, usually that indicates that the matrix gene is present. With the albino, they all have pink tongues as well because, that reduces that pigmentation. So that makes it a little bit more difficult to tell. At that point, you go off the pattern and you get more and more experience and used to what you're looking at, and you'll be a lot better and make more predictable results. It's always easier, though, if you have an ivory involved in the pairing, and then for certain matrix is present, there's no question. When you do matrix to matrix, you can get those ivories, but you can also get those wild types that don't have one allele or both alleles for ivory. So hopefully that'll help you a little bit. Hopefully I haven't created more confusion. Feel free to ask any questions on the video or anywhere that you see this, and I can try to explain a little further. I'm hoping that you watch it a couple times, maybe it'll digest a little better. And if you have individual, you really want to explore more, because there's a lot of terms I left out to simplify this, when we get into mitosis and meiosis and all that kind of stuff, and how the cells divide and how these things are created. There's a lot more that you can research here, but I wanted to do something that kind of stayed under 20 minutes and gave you a little crash course, at least on blood pythons, of how some of these modes of inheritance work. So you saw recessive genes and you saw incomplete dominant genes. Now, if batik was simply just a dominant gene, the same results would happen when you bred it to a normal. You would get some batiks and some normals, but that's the end. It's not an incomplete dominant. There's nowhere to go from there. You don't breed two batiks together and get something else. You would just get more batiks. So that's the difference between dominant and incomplete dominant. And I'm not sure if bloods have dominant traits. I don't work with any, so I'm not 100% positive. I know in ball pythons there are some, though. So hopefully that helps you guys out. Any questions, let me know. I'll get this little guy put away and wrangle the other ones that made this video a little bit of an adventure. Thanks, guys.